Okay, shall I start, Dr. Esteller?
Yes, so just to say welcome everyone. Sorry for the delay due to technical problems. It's not COVID related. We cannot blame COVID for this. Just to say a great pleasure to have with us Professor Swanton. He combines his laboratory research at the Francis Crick Institute at UCLH and also as director of the Crick UCL Lung Cancer Center. He's a fellow of the Royal College of Physicians, Academy of Medical Science, and Royal Society, and also a recent fellow of the American Association for Cancer Research. He has received many awards for his research. Among them, I would like and recognitions like Stand Up to Cancer in 2015, the Gordon Hamilton Fairley Medal 2018, the Mass General Hospital Kraft Prize 2018, the ESMO Award in 2018, etc. And all these recognitions are for his excellent work in the area of, in many areas, but I really like the area of branch evolution and immune evasion associated with these different arms and branches in cancer, and mainly in lung cancer. So it's a great pleasure to have with us Professor Swanton.
Thank you very much indeed for the very generous introduction and your kindness bearing with me through our IT problems here at the Francis Crick Institute. I am sorry. So I'm going to quickly talk to you today for the next 40 minutes or so about chromosomal instability, which we think is a major and rather under-recognized driver of cancer evolution, immune evasion, and metastasis. Here are my financial disclosures. So we set up TracerX about 5 years ago to understand the patterns of evolution from primary diagnosis through to recurrence and ultimately death. And the idea is that through extensive tumor sampling of the primary tumor, we can help to distinguish the metastatic competent from incompetent subclones in the same patient through multi-region sequencing of the primary, matched with recurrence sequencing of the metastasis, and ultimately, through some of the cases, autopsy analysis of multiple sites of metastatic disease. The endpoints of TracerX are shown here. Broadly speaking, we want to understand the relationships between genetic diversity and outcome. Are there evolutionary rule books which we can predict, and can we time events during evolution? And what are the origins of the lethal metastatic subclones? And can we use ctDNA analysis to understand cancer evolution? And ultimately, can we infer mechanisms of branch evolution and immune escape? So before I get on to chromosomal instability, I want to talk to you about microevolution. That's small incremental change over a long period of time brought about by point mutations. These are the three main point mutational processes we see in lung cancer: the tobacco carcinogen signature, the C2A mutations, ApoBec C to T mutations, and spontaneous deamination mutations, C to T mutations. When we look at those signatures in terms of the early versus later events in tumor evolution and focus initially on the early events in tumor evolution, what we see here are the smoking mutations dominate the trunk of the tumor's evolutionary tree. That is, the number of pack years that patients have been exposed to in lung adenocarcinoma directly correlates with the number of C to A mutations in the trunk of the tumor's evolutionary tree. Now, if we ask what mutations are active at the point of diagnosis, even in a smoker, the smoking mutations are no longer significant in the branches, but now we see the mitotic clock and ApoBec cytidine deaminase mutations dominate the branch mutational signatures. So that means even in a smoker, the most active mutational processes at diagnosis are these C to T mutations brought about by the aging signature and ApoBec cytidine deaminase signature. So as a medical oncologist, what does this mean to me? Well, what I think we infer from this is that precision oncology is going to be a difficult task for outside the sort of EGFR, BRAF, HER2 mutations that you'll be familiar with. And for the squamous carcinomas particularly, what we're dealing with are very heterogeneous diseases with an array and panoply of different driver mutations, both trunk and branch. And of course, these are underpinned by different genome instability processes. You can see here from the first 100 branch trees that we see in these patients. So what about the first clinical endpoint of TracerX, the relationship between genomic diversity and cancer outcome? What we see here is that point mutational diversity is not associated with poor outcome. Instead, chromosomal instability, that is the diversity of somatic copy number aberrations between subclones in the primary tumor, is associated with poor outcomes. So patients with high chromosome instability have poorer clinical outcome than patients with lower chromosome instability. And this harks back to the evolutionary biologist Richard Goldschmidt, who coined this phrase, hopeful monsters, referring to karyotypic change during evolution as a means to speciation. And rare karyotypic rearrangements can result in profound change and phenotypic advantage. So the question is, do these somatic copy number aberrations result in neutral growth, or are they positively selected throughout tumor evolution? And I'd like to refer to work that people in my lab have been doing, along with Nikki McGranahan, a talented independent principal investigator in the lung center that I run, who's been helping me lead this analysis with Roland Schwartz at MDC in Berlin and Sam Bakum at Memorial Sloan Kettering. And to do this, what we did is we obtained really the global resource of multi-region data sets sequenced through exome or whole genome sequencing, and in some case SNP profiling, and subjected 1,421 regions from nearly 400 tumors across 22 tumor types to a common informatics pipeline to understand the role of late and early somatic copy number aberration events in tumor evolution, conducted by two talented postdocs, Tom Watkins and Amelia Lim. And when we look at this across the genome, we take a tumor like squamous carcinoma of the lung, what we see is, a very standard copy number plot that many of you will be familiar with, overlapping many familiar tumor suppressor genes and oncogenes. But now with our new tools, we can break those down into whether they're early events or late events. And what we see is that across the genome, we see these plots divided by whether or not these copy number gains and copy number losses occur early or later on in tumor evolution. In blue here, you see the subclonal copy number gains and losses. And what we infer from this is you can see early losses in the context of p53, CDKN2A, and chromosome 3p in squamous carcinoma of the lung that you'll be familiar with, that are almost exclusively early in evolution. And then you see later losses that are almost exclusively late in tumor evolution, including chromosome 7p gains overlapping EGFR and CDK6, which in squamous carcinoma of the lung are almost always later events in tumor evolution, along with many others. And then there's some events like chromosome 1q that can be either early or late. So what we infer from this in the pan-cancer analysis is that there is distinct order to this chaos of copy number aberrations in cancer evolution, with early events overlapping familiar tumor suppressor genes, intermediate events overlapping with many oncogenes you'll be familiar with, including MYC and cyclin D1 and cyclin E1, and later events, including HLA, loss of heterozygosity, and SMARCA4 copy number gains. And what's intriguing about this is that these events vary between tumor types. So for example, in 7 out of 13 tumor types, CDKN2A loss is an early event, CYP-D2 loss is an early event in 8 of 13 tumor types, and 17p13.3 loss encompassing TP53 is an early event in 9 tumor types. But in other tumor types, for instance in breast cancer, we find that, for instance, FAT1 loss is early in triple negative breast, but a late event in HER2 positive breast cancer, and intermediate in ER positive breast cancer. But as I mentioned earlier, HLA-LOH is late in 8 out of 13 cancers. So it's late in almost all cancers that we study as a subclonal event when it occurs. So next we sought evidence of selection through looking for evidence of parallel evolution. And in copy number space, we refer to something called mirrored allelic subclonal imbalance, a phrase we coined in 2017 when we first started looking at this in lung cancer. This is where maternal and paternal haplotypes are amplified in distinct subclones of the tumor. So let's say you could have HER2 amplification in region 1 of the tumor from the paternal allele, and HER2 amplification from the maternal allele in region 3 of the tumor. So nature will ultimately find a way to find a solution to the same problem through amplifying up HER2 from either the paternal or maternal alleles. And intriguingly, we see a lot of evidence for this across tumor types in our pan-cancer analysis. In 37% of tumors studied, we find one or more parallel somatic copy number aberration events. So you can see here, for example, MYC in this tumor, CRUK0070, is gained twice independently from the maternal allele in one set of subclones and the paternal allele in another set of subclones. And the same can happen for HIF1 beta or GNAS, and it can occur for losses too, including P10, CDKN2A, and HIF1 alpha. And you can see here, we see HIF1 beta copy number gain and HIF1 alpha copy number loss later on in tumor evolution, suggesting convergent evolution upon hypoxia signaling later on in evolution. Finally, we sought whether there was evidence of selection by modeling what's called the oncogene and tumor suppressor scores from Teresa Davoli and colleagues and Steve Elledge in Boston. And this essentially sums up the proportion or the number of oncogenes compared to the number of tumor suppressors on any one chromosome arm, postulating that you would select for chromosome arms with a greater proportion of oncogenes and a lower proportion of tumor suppressor genes. And when we apply that algorithm, we see that really there's the same level of selection for clonal copy number gains and losses as there is for subclonal copy number gains and losses, so suggesting that growth is not neutral in the branches and that these copy number events at the arm level at least are selected as tumors continue to evolve. So in summary for this part of the talk, for this paper that we published a couple of weeks ago that you can find more details of this year, is that we think that there is quite significant evidence for ongoing positive selection during tumor evolution of somatic copy number aberrations. But the question is, how is SIN generated? How is chromosomal instability generated? This high-risk phenotype, or genotype, I should say, or karyotype in non-small cell lung cancer. Well, intriguingly, we find that genome doubling is a very common event in tumor evolution, and we refer back to seminal work from David Pellman and colleagues, who first postulated in 2005 that genome doubling was a high-risk phenotype in cancers. He showed that genome doubled mouse mammary epithelial cells are very much more aggressive than the non-genome doubled counterparts. In TRACERx, we find that genome doubling occurs in nearly 75% of patients, and when it occurs clonally in the trunks in almost all patients studied. We've also been studying genome doubling for several years now, and we've modeled this in vitro in the lab in isogenic cells. And when tumor cells double their genome over 18 months in culture, these are 6 different genome double clones derived from their parental diploid counterparts, they become very much more chromosomally unstable over time compared to their diploid counterparts who remain robustly diploid over the course of the 18-month experiment. When we look at TRACERx, we find that tumors with genome doubling are very much more chromosomally unstable than their non-genome double counterparts. So genome doubling, we would argue, is not only associated with chromosome instability, it drives chromosome instability in one way or another. But that doesn't explain why it is selected. And so this is what I want to focus on for the next 10 minutes or so, and focus on some work that Nikki's been leading in the lung center, which really alludes to this quite elegantly, I think. And so this work sort of harks back from Julian Huxley's analysis back in 1942, in the Second World War, when he suggested that tetraploid forms are in many cases more vigorous, reflected in their wider distribution than that of the diploid variety. And I look at that quote and I think of a metastasizing tumor. And we've been very interested, really, in the last 7 or 8 years in why genome doubling occurs, and most importantly, why it's selected. And this dates back to a conversation I had with Nikki when he was a PhD student in my lab, where we postulated that maybe genome doubling was being selected because of a Muller's ratchet-like phenomenon. And Nikki's sort of taken it upon himself to analyze this phenomenon really for the last sort of 5 years and into his own independent lab, where I think he's begun to shed light on the fact that this is indeed possibly correct. So the concept of Muller's ratchet is that in asexually recombining organisms, or sorry, in asexually reproducing organisms with no recombination, the progressive accumulation of deleterious mutations ultimately leads to death. Now, in sexually recombining organisms, we have the luxury of recombining out these deleterious mutations to result in mutation-free chromosomes and survival. So we reasoned that in a diploid organism that has an essential gene, let's say, that becomes haploid due to chromosome instability, which we see in lung cancer, that leaves the emerging tumor cell with a bit of a problem, because if it gets a secondary hit in that remaining single copy of that essential gene, the cancer cell will die. So we asked, is there evidence for purifying selection of deleterious mutations in essential genes which are down to 1 copy in regions of haploid LOH that we see before genome doubling, and we no longer see evidence of purifying selection after genome doubling, by virtue of the fact that there is still a remaining allele left in the tumour that can function appropriately. And to do this, we used the Martin-Karenner approach, the DNDS ratios borrowed from population genetics, with a DNDS ratio of 1 inferring neutral evolution, DNDS ratio of greater than 1 inferring positive selection, and less than 1 inferring purifying selection. And applied that to TracerX analysis, where we took the essential genes from CRISPR screens that are in the public domain and asked, is there evidence of purifying selection before genome doubling in these essential genes that you don't see after genome doubling? And that's exactly what you see, that in regions of haploid loss of heterozygosity, when these essential genes are down to 1 copy, there's very profound evidence of purifying selection or negative selection against further mutations in these genes that you no longer see after genome doubling. So that sets us on a path to ask whether there might be a relationship between tumors that double their genomes and the proportion of those genomes before genome doubling that are subject to haploid loss of heterozygosity. And that's exactly what we see. There's a very strong pan-cancer relationship between the proportion of tumors with double genomes and the proportion of those tumors that have their genomes subject to haploid LOH. So in other words, tumors with very high proportion of the genome subject to haploid LOH, like squamous carcinoma of the lung or triple negative breast cancer, these tumors have a very high frequency of genome doubling. In contrast, for instance, microsatellite unstable colorectal tumors, which have a very low frequency of haploid LOH, also have a very low frequency of doubling of their genomes. So next I want to talk about the impact of chromosomal instability on deciphering the immune landscape and actually immune evasion specifically. And this is work conducted and led by Rachel Rosenthal and jointly supervised by myself and Nikki, and really to try to understand how lung cancers evolve over space and time in the context of a predatory immune system. So when Rachel and Nikki looked at the TracerX data, what they saw is essentially 3 types of tumors. You can have tumors that are exclusively hot. So they have, whichever tumor region you look at, they are hot. They have brisk lymphoid infiltrate. Now, there are tumors that are mixed, where you have some regions that are hot and some regions that are cold. And then you have some tumors that are exclusively cold. Whichever region you look at, there's no evidence of a brisk lymphoid infiltrate. So now we've classified those different tumors into whether they're hot or cold or mixed, we can now start to ask what potential mechanisms of immune evasion there may be that relate to cell intrinsic mechanisms of immune evasion, shown here. So we can have HLA loss of heterozygosity, antigen presentation disruption, or neoantigenic evolution in the context of copy number loss of neoantigens. So when we first of all look at HLA-LOH, using a tool that Rachel and Nikki developed called LOLA, we find that HLA-LOH is present in about 40% of non-small cell lung cancers in the early stage setting. Now, I should just reiterate and emphasize that none of these patients has had any chemotherapy or immunotherapy treatment. So these are chemo-naive, immunotherapy-naive, early stage lung cancers. In these tumors, we find in a prospective cohort of 100 patients, nearly 40% of tumors have lost 1 or more HLA class 1 allele. Next, we ask, when does this occur? And we find that it occurs commonly as a subclonal or branched event during tumor evolution. More commonly, in fact, than it does as a clonal event during tumor evolution. So interestingly, it's a later event. And we still don't know the answer to that, why that is. But we suspect it may reflect the underlying bulk of disease and the number of tumor cells and the clonal fraction of neoantigens that are being presented to the immune system, and may also start to explain why you don't see lymphoid infiltrates in normal tissue as a result of normal somatic mutagenesis. So here's some examples of HLA-LOH as a truncal event. And here's some examples of HLA-LOH as a branched event during tumor evolution. And here's some intriguing examples of parallel evolution, where the same HLA allele is lost on more than one occasion during tumor evolution. And I'm zooming in on those 4 tumors here. You can see that the same HLA alleles are lost recurrently on 2 different occasions in 2 separate branches of the tumor's evolutionary tree. And here you can see this peak in chromosome 6p overlapping HLA, really providing quite strong evidence that HLA LOH is selected for as a later event during tumor evolution through chromosomal instability. So next we were interested in understanding what impact that might have on tumor evolution. And what we find is that if you track subclones in TRACERx and you look at subclones that have lost HLA versus those that haven't, what we see is the subclones that have lost HLA have more mutations in them than those that haven't. They're sister subclones in the same tumor that haven't. They're as closely matched as possible. So this suggests that losing HLA allows tumors to now evolve below the radar, if you like, and evade a predatory immune system and acquire mutations without being recognized. So next we ask, where are these HLA events occurring? So they're occurring in tumors by and large that are hot. So we see HLA-LOH in immune hot tumors or tumors that are immune mixed, in general, those with a brisk lymphoid infiltrate, and less commonly in the immune low tumors. We see HLA-LOH in tumors with high PD-L1 expression, again, consistent with this idea that they're immune hot and immune activated. And intriguingly, we see a trend towards HLA-LOH at the primary to metastatic transition. This is data from Brastianos and colleagues, where they have primary and central nervous system metastasis in non-small cell lung cancer. And we see a trend towards loss of HLA-LOH during that primary metastatic transition. So that's immune evasion in immune hot tumors. HLA-LOH seems to be a dominant mechanism, but we also find that antigen presentation machinery, copy number loss, and mutations is a common mechanism too in immune hot tumors that tends to occur mutually exclusively with HLA-LOH. So we see that, for instance, even in specific tumors, we see that you either get HLA-LOH or antigen presentation disruption machinery mutations or copy number loss events, but less commonly both occur in the same region. And when you take this as a whole, between 56 and 78% of tumors, depending on whether adenocarcinomas or lung squamous carcinomas, have HLA-LOH or antigen presentation machinery disruption. So there seems to be considerable evidence for immune evasion in early stage untreated non-small cell lung cancer. So I now want to briefly move on to talk about what we're doing clinically to try and overcome this challenge. And what we've inferred from years of clinical practice and success in the field from colleagues across the world in the targeted therapy space, is that we're very good at targeting single point driver mutations that are almost always clonal in nature. So these early founder initiating events like EGFR in non-small cell lung cancer, we're very effective at treating, with the vast majority of patients responding to therapy at all metastatic sites of disease commonly. Not always, but commonly. But the problem is resistance to therapy is almost inevitable, brought about by new or previous subclonal mutations that occur as resistance events during tumor evolution. So what do we need to do? Well, I think what we need to do here is we need to tackle multiple clonal mutations during evolution to prevent resistance from occurring. But how are we going to do that, given every patient has a unique phylogenetic tree, as I said to you earlier? Well, intriguingly, in non-small cell lung cancer, in smokers have a very high clonal mutational burden. So they have a coding mutational burden that is about 5 to 10 times higher than in a never smoker. So ex-smokers, recent smokers, or current smokers have about 500 coding mutations in the trunk of their tumor's evolutionary tree, as opposed to sort of 50 to 75 coding mutations in a never smoker. And this suggests that there are many more potential neoantigens that one could prosecute and target with the benefit of T-cell or vaccine therapies. So how important are these clonal mutations? So we looked at this in the context of anti-PD-1 therapy, as well as more recently a meta-analysis of about 1,000 patients, which I'll show you in a minute, to ask the question, are clonal mutations more important than branch mutations in conferring response to immunotherapy? So when you think about tumor mutational burden, is it the clonal mutations or the branch mutations that confer sensitivity to immunotherapy? And what we find is that it seems to be the homogeneity of the tumor and the truncal mutational burden that seems to best correlate with response to checkpoint blockade. This is a small group of PD-1 responders versus non-responders, where you can see the branch mutational burdens enriched in the non-responders. And this was subsequently validated by a friend and colleague, Ellie Van Allen, in Boston, who showed in a cohort of 244 patients that the branch mutational burden conferred poorer responsiveness to checkpoint inhibitor blockade. Kevin Litchfield in my lab has been working for the last 3 years on this problem through building a meta-analysis of about 1,500 patients treated with checkpoint inhibitor blockade, breaking down mutational burden into its component parts, clonal and subclonal. And what he finds is it's the clonal mutational burden in this meta-analysis of about 1,000 patients treated with immunotherapy across multiple tumor types that appears to be the strongest predictor across all the potential biomarkers that have been published in the literature in the context of response to anti-PD-1 and anti-CTLA-4 therapies. Interestingly, subclonal mutational burden does not seem to correlate with response to therapy. So this got us thinking, is it that clonal mutations are important and that subclonal mutations just aren't? Or could subclonal mutations actively be preventing response to therapy? And we were lucky enough to collaborate with Yardena Samuels at the Weizmann Institute, who'd set up a very elegant animal model, who was addressing this question very rigorously in the lab. And I'd like to tell you a little bit about her work, because I think it shines a very bright light on this challenge of immune control and immune failure. So we worked with Yardena on the bioinformatics front. She did the lion's share of these studies, and she's the corresponding author on this paper, quite rightly. And she developed this syngeneic system that many of you will be familiar with, the B2905 mouse melanoma model, which she exposed to UV irradiation and single cell cloned to create diverse phylogenetic trees, very clonal ones and very subclonal ones, to ask what impact would that have on spontaneous immune rejection of the B2905 syngeneic melanoma line in a mouse with a competent immune system. And she did this by varying the proportion of single cell clones that she put together and injected into the skin of a mouse to stimulate immune rejection. And what she finds is, to cut a long story short, and this work was published last year in Cell, that the more complex the tumor phylogenetic tree, the less well it's rejected. And the more simple the phylogenetic tree with the more clonal mutations, the more swiftly it's rejected. And she inferred from this that it's the number of clones and their genetic diversity that play an important role in mediating tumor growth and rejection. In other words, what we take home from this is that the tumor branches, or the heterogeneous branches, can actually deflect the immune system from what's really important, which is the truncal mutations. So now let's go back to the bedside, and I'd like to tell you briefly about what we're doing in the clinic. And this is where one of my conflicts of interest comes in as a co-founder of a company called Achilles Therapeutics. Here, we're using T cells to target multiple clonal mutations. And this builds on work from Rachel Rosenthal, Nikki, Andrew Furness, Sergio Quesada, in collaboration with Sine Hadrup in Denmark, where we use Sine Hadrup's technologies built with Ton Schumacher, this tetramer approach, which are these synthetic HLA tetramers, which are synthetic HLA-MHC peptide complexes that can be used to fish out of tumors T-cell receptors, T-cell alpha-beta receptors, that specifically bind and interact with clonal neoantigens. And we've done this across many tumors now, and in many of these, we can find multiple different T cells that recognize multiple different truncal neoantigens. And what we're doing now, we set up 2 trials in lung cancer and melanoma through this company, Achilles Therapeutics, which I have a conflict of interest in, to treat patients with T-cell therapies, specifically recognizing truncal neoantigens. Now, I'm not involved in those trials, obviously, but I'm very excited to see what the outcomes of those trials will be, with the idea that we can offer back to a patient, having grown ex vivo multiple different T-cells against multiple truncal antigens, T-cells that specifically recognize truncal mutations, multiple truncal mutations that we hope will prevent resistance from occurring. So now I want to come back, in light of this discussion around clonal neoantigens, to how immune cold tumors are evading treatment, or how they may have evaded, I should say, not treatment, how they evaded a predatory immune system in years gone by. And I see this as sort of a tantalizing fossil record, and I'll explain why. About 4 years ago, when we were looking at TRACERx data, what we saw is something we called an illusion of heterogeneity. What that means is that you can have a clonal or truncal mutation shown in a star here that was once upon a time in evolutionary history truncal or clonal in nature, but by virtue of chromosomal instability and a copy number loss event of a chromosome arm or a small segment of a chromosome has been deleted. So this truncal mutation is actually now heterogeneous in the tumor mass. And we wondered why this was happening. And it was only until we made this foray into immune evasion that we reasoned that it might be happening due to copy number loss of neoantigens. And that's exactly what we see. So that in immune cold tumors, we see significant increase in copy number loss of neoantigens that we don't see in immune hot tumors. So what we reason from this is that perhaps this is a fossil record. This is telling us that in years gone by, these tumors might have been hot, and there was a selection pressure for loss of these neoantigens or some other immune evasion mechanism. And in that particular region, loss of clonal neoantigens was selected, and as a result, there was no longer an immune signal, and presumably the T cells have disappeared over time. Now, of course, we've got no evidence for that other than this fossil record, but that's what we reasoned from this analysis. So in summary, we see all 3 mechanisms of immune evasion differentiated by whether they occur in immune hot or immune cold tumors. And then we take this together in the first 100 patients. What we find is that patients that have no evidence of immune evasion do very well, and they have a very good disease-free survival in multivariate analysis that is independent of tumor stage or other prognostic factors. So when thinking about this more carefully, what do we take home? Well, I think targeting neoantigens is going to be challenging, not least of all because very few neoantigens will be in driver events. What does that mean? Well, it means that the tumor can find multiple ways of rewiring or re-engineering the genome to ultimately evade the immune system at no threat to itself. So take copy number loss of neoantigens as an example. So we need to think more cleverly. We need to think what Darwin would do in this situation. Well, what I think Darwin would do in this situation is he would try and constrain the system so that, or to stack the odds in the patient's favor to prevent neoantigens being subverted or deleted or repressed, or to prevent the impact of HLA loss upon immune evasion. So what should we do? Well, first of all, I think it would be sensible to find neoantigens that bind multiple HLAs so that losing one HLA doesn't result in failure of antigen presentation. How do we do that? Well, frame shift insertion deletion antigens I haven't had time to show you. Kevin Litchfield has shown this very nicely in Nature Communications in my lab a couple of months ago, that neoantigen copy number, frame shift insertion deletion mutations bind to multiple HLAs. Now, another thing is obviously clonal fraction. So neoantigen clonal fraction is important. That is, the clonal fraction of mutations is likely to be important to confer optimum immune recognition, and that branch mutations may even deflect the immune system from what's important, the truncal clonal mutations. I mentioned to you that neoantigens rarely occur in driver genes, but there's another way of looking at this, that maybe they occur in genes required for cell viability, so-called essential genes. And I showed you earlier the importance of essential genes in tumor evolution in the context of genome doubling. Then there are other aspects like the similarity to self that may impact antigen recognition and the likelihood of loss, looking at areas of the genome that are less frequently subject to copy number loss. And of course, we want to select neoantigens that are expressed, and of course that will tie into their role in the context of cell viability. So in summary, I would like to say that I think chromosomal instability is a very important factor in tumor evolution, in that it remodels tumor ancestries. It's associated with poor prognosis. It permits immune evasion through clonal neoantigen loss and HLA loss of heterozygosity. And one thing I wanted to point out, this occurs after genome doubling very commonly. Now, one thing I didn't mention, but may have occurred to many of you in the audience, that when you double the genome, you now have 2 copies of HLA. And the fact that we see HLA-LOH in genome double tumors is extraordinary, because what it tells you is that 2 copies of that same HLA allele are being lost, telling you that there is clearly a profound selection pressure for HLA-LOH during tumor evolution. And of course, chromosomal instability creates the diversity upon which selection can act during tumor evolution. We see parallel evolution of copy number events. I've shown you evidence for MYC, HIF, and the catalytic subunit of TERT, telomerase. Ongoing copy number gains occur in oncogenes later on in evolution that we see in the context of our selection models and selection of chromosome arms with oncogenes versus those that are depleted of tumor suppressor genes. When we look at cancer evolution as a whole from TRACERx, what has it taught us? Well, what we've seen is that clonal drivers early in tumor evolution are very familiar to us. EGFR, KRAS, P53, for example. Tobacco carcinogens dominate these truncal mutations in smokers and ex-smokers, and it's those by and large that we're hoping to target in the context of this new era of T-cell clonal neoantigen targeted therapies, leading to a high clonal neoantigen burden. But chromosomal instability occurs early in tumor evolution, providing the substrate upon which diversity acts. You can have HLA-LOH occurring early and late, but genome doubling is almost exclusively early, and we think occurs because of this problem of chromosomal instability around essential genes in tumor evolution, creating this very high proportion of the genome that's subject to haploid LOH. Roughly 30–35% of the genome is subject to haploid LOH before a genome doubles in squamous carcinoma of the lung, one of the highest of all tumor types. Then you get multiple mechanisms of immune evasion shown in green, chromosomal instability driving parallel evolution, and cytotoxic and ApoBec C to T mutational signatures. I'd argue that whole genome doubling is a transformative event in tumor evolution. It solves the problem of Muller's ratchet, and it's associated with further chromosomal chaos, chromosomal instability, that provides the tumor with many more avenues to explore, and presumably the diversity upon which natural selection can act. So in summary, I'd like to thank everybody for making this possible. This has been a huge amount of teamwork. This isn't me, this is many, many talented scientists in the lab. I've tried to name many of them during my talk, but I've not been able to mention even a small proportion of them. Mariam Jamalhanjani, Nikki McGranahan, Gareth Wilson, Chris Abosh, Nikolai Birkbach, Rachel Rosenthal, Tom Watkins, Amelia Lim, Kevin Litchfield, amongst many others. TRACERx, Cancer Research UK, Andrew Rowan for being at my side and leading the lab really from its inception. The team, patients and their families, Nikki McGranahan's lab, Kevin's lab, and TRACERx lab across the country. I'd like to thank the Royal Society, Cancer Research UK, the Francis Crick Institute, UCL, and the hospitals and UCL hospitals for making this work possible, and of course to Dr. Esteller and your colleagues for inviting me today. I hope I can answer some of your questions, and I'm sorry for the technical problems we had early on in the talk. Thank you.
Thank you very much. Dr. Swanton was great, really great. I'm very condensed, very intense. So thank you very much. So I have questions from people attending here. I'm trying to organize them a little bit. One is very general. Is according to your data, to this data and data for others, there are immune cold, immune hot, etc. So do you think at the end everybody is going to receive immunotherapy? Or really the biomarkers in lung cancer are necessary?
Yeah, I think biomarkers are going to be essential. I think it's like everything in cancer medicine that you know, it's incremental change over a long period of time that makes a big difference. I don't think, you know, I think immunotherapy is extremely exciting, and I think it's going to be fantastic, and it is fantastic for a proportion of patients. But I don't think it's going to be a one-size-fits-all approach for everyone. And there are going to be some tumors that just by a quirk of evolutionary fate will not be immunogenic. I'm pretty confident about that. And biomarkers are going to be absolutely essential to tease apart that group of patients for which immuno-oncology approaches aren't appropriate. What those will be, I think only time will tell. Tumor mutational burden may be a part of that. I don't think it's the whole story. We estimate that the biomarkers we currently have in existence, clonal mutational burden plus others, account for about 60% of the variance. So there's still about 40% of the variance of response to checkpoint inhibitor therapies that we still haven't explained. So there's a lot of dark matter that we don't know about.
Other relates that what happens with natural killer cells
Yes,
in your models of HLA-LOH?
So we're just beginning to look at that in the context of a microenvironmental study. What we find is evidence in tumors that are heterozygous for HLA-C1, C2. If you lose one of those alleles, C1 or C2, you get NK cell infiltrates. That's really where the only place that we see a strong NK signal. We don't see an NK signal quite so strongly with other loss of other HLA molecules. So at the moment, it just seems to be an HLA-C specific event in lung cancer. That may be a sensitivity issue, I don't know.
And the other relates about how universal are these findings. For example, you studied a lot the lung cancers. In phenotype-driven tumors from melanoma and colon, do you think it's the same model? It can happen the same? It's a different profile of the branching?
Yes, absolutely. I think branch evolution is universal. I think what we see here, I think lung cancer, it just happens that lung cancer, the disease I treat and study, is at the interface of mutational diversity and chromosomal instability. It's one of the tumor types with our greatest burden of somatic mutations and the greatest proportion of the genome subject to chromosomal instability. Other tumor types will be similar from each perspective, but I don't think, you know, most tumor types won't be as extreme as lung cancer. So to give you an example, HLA-LOH occurs rarely in melanoma. It occurs in only about 4% of patients, whereas it occurs in about 40% of lung patients. And the reason for that, we think, is that there is an oncogene on chromosome 6p, very close to a lineage-specific oncogene, very close to HLA called NED9. And that cannot be lost during tumor evolution. And clearly that presumably constrains melanoma, preventing copy number loss of HLA during tumor evolution. So I think the principles here broadly will be translatable across other tumor types, and indeed are. But the degree to which you see each across individual tumor types will vary.
And the other relates in those patients with carrying the EGFR mutation, they are less heterogeneous?
No, that's a good question. A lot of this comes down to how you measure heterogeneity. So if you look at the proportion of mutations, some people argue actually that the proportion of mutations in EGFR mutant disease is greater subclonally than clonally. I would argue what the immune system sees is the total number of mutations rather than the proportion. And it's that matters. And if you look at that, it is indeed correct that if you look at the total burden of subclonal mutations, smoking-related lung cancer is very much more heterogeneous than EGFR mutant lung cancer.
In the interest of time, I will select just a couple more, and I will leave you free.
Cool.
So, do you think that assessing only clonal status based on BAF and the frequency of the altered allele is too risky, or do you think this is a useful biomarker?
No, you can't. It's too risky. It's a difficult thing to do. You need to correct for tumor purity and local copy number. You can't. It's very hard to do based on VAF for the simple reason that many of these oncogenes, for example, are subject to copy number amplification. So you can have a subclonal oncogenic driver that is amplified that appears at VAFs that are very high indeed, that you would be mistaken for thinking are clonal in origin.
Yeah. And you said that many of these findings can be expanded to other tumor types. But what about tumors in kids that they said that they have more alterations in development? Can be different, or they are the same?
I perhaps shouldn't have said been quite so widespread in my assumptions. I think pediatric tumors are a very special case. They have very low mutations, as you know. Some of them are very chromosomally stable too. But actually, interesting, there are still quite a considerable proportion of pediatric tumors that have a degree of chromosomal instability too. I'm pretty convinced that chromosomal instability is critical, particularly in pediatric tumors where there are no obvious driver mutations. Chromosome instability is likely to turn out to be an important facet of tumor evolution in those tumor types, in some of those tumor types at least.
And we'll finish with one question about cellular therapy. It's a very interesting issue for many people here also. Let's say in the TILs, in your T-cell therapy, this is given in the blood, or this is local therapy in some way? And how do you see the future of CARs for solid tumors?
Yeah, so I think they'll be given intravenously. Well, they are given intravenously, not locally. And because they're taken from the tumor, the principle is that they will track back to the tumor. So that you take a tumor biopsy to access the T cells, ex vivo expand them and then give them back. And the idea is that they'll have their homing devices intact. Obviously, this all remains to be proven, but that's certainly what's been seen by Rosenberg and others. The CAR T-cell therapy, I think I'm less optimistic about CAR T-cell therapy in solid tumors for the simple reason that we really lack tumor-specific lineage markers that you can safely target. So, you know, we've been carried away to some extent by the ALL field because of the fact that humans, fortunately, can live without the B-cell compartment. So you can target CD20 as a sort of lineage-specific target, and apart from requiring regular antibiotics and what have you, and immunoglobulin, you can live without B cells quite well. Whereas in lung cancer, you can't live without your lung epithelium, obviously. So the question is there a lineage-specific marker that can mark out cancer cells, cancer epithelial cells, that won't result in targeting the lung epithelium and leave the lung epithelium and other epithelial tissues for that matter intact? And I think that's the challenge. And I'm less optimistic about that. I'm more optimistic about a future where you have vast warehouses full of engineered T-cell receptors that can be specifically matched to the patient's own HLA and a neoantigen of choice. I think that's more likely.
Yeah. Okay, in the interest of time, I think we should finish. I would like to thank you very much for your participation. Sorry for the late delay in the meeting.
I'm sorry.
And it has been a real pleasure. Thank you very much.
Thank you very much indeed. Thank you.
Take care.
Thank you.